Good evening, everyone. I'm Noelle Osen, President of the Kalamazoo Area Wild Ones, and I want to welcome you this evening to the first in our 2025 series of programs. 2025 is going to be another exciting year for KWO. We'll be continuing with our extremely popular Pints and Plants monthly gatherings at Brewery Utra in downtown Kalamazoo. There are also going to be field trips and garden tours in our annual plant sale. I hope to see many of you at a lot of these events during the course of the year. Now I'll turn the program over to Quinn Edwards, who will introduce tonight's speaker.
Thanks so much, Noelle. Good evening, everyone. My name is Quinn Edwards. I am the chapter's vice president this year, and I also sit on the KWO programming committee. I've been looking forward to this program for a long time now, so it's my pleasure to introduce you to tonight's speaker. Ashley Cole-Wick received her undergraduate degree in environmental science and policy and a master's degree in conservation biology. Ashley joined Michigan Natural Features Inventory in 2018 with a mission to protect our state's rare species like the Mitchell's satyr and Karner blue butterflies. While at MNFI, she has also conducted rare species and ecological inventories on tribal lands and has conducted surveys for aquatic plants, small mammals, land snails, freshwater mussels, aquatic snails, and various insect species. Ashley enjoys helping people better understand the natural world around them so that they can work to protect it. And with that, I will turn it over to Ashley.
Thank you. Yeah, it's great to be here, and I recognize so many names from around town and also my previous position at the Kalamazoo Nature Center. And I'm going to go ahead and share my screen and get started with this presentation. So can everyone see that?
Looks good.
Yes.
Great. Okay, sorry, I have a couple little puppies with me, so they're making some noise, but we'll forgive them. Okay, so tonight I am going to share with you all some work about some work with a couple endangered butterfly species here in Michigan. On the left, we have the Karner blue butterfly, and on the right, we have the Mitchell's satyr butterfly. And I will end with a tiny bit about the Poweshiek skipperling. So those three species are our three rarest butterfly species here in Michigan, all with federally endangered status. And yeah, like was mentioned in that lovely introduction, thank you. I'm a conservation associate and zoologist at Michigan Natural Features Inventory. I'm going to start out our talk today with a little bit about MNFI. I'm sure many of you know about our organization and the work we do and have heard from my colleagues, but just a little bit about that. And then we're going to get into the work we're doing with the Mitchell's satyr, and I'll go ahead and get started. So a lot of times people finish presentations with acknowledgements, but I like to put mine up front here so that I can make sure I really dedicate a moment to all the people who help make this work possible. I'll say the royal we a lot during this talk, and not only my colleagues at MNFI, but even pictured here, we have folks from Camp Friedenswald, the DNR, Southwest Michigan Land Conservancy, the Kalamazoo Nature Center, the USGS, Fish and Wildlife Service. So the work on these species is really a joint effort between so many folks. So I just want to shout out a thank you to all of them. So Michigan Natural Features Inventory, that's our mission statement I put up there. Guide the conservation of Michigan's biodiversity by providing the highest quality scientific and expertise and information. So A big part of what we do is we manage our state's natural heritage database. Every US state, every Canadian province, and many countries in Central and South America have very similar organizations. And we all contribute data not only to the Michigan database, but then the national database. So when you see that a species is listed as G1, globally imperiled, or S1, state imperiled, these are the data that go into making those decisions. So when a species with the DNR or Fish and Wildlife Service gets uplisted to a more from threatened to endangered, or it gets downlisted, we help contribute the data to make those decisions. And at the end of the talk, I will have a little bit of a call to action on how you all can help me and help us do our work. And a part of it is you guys being out here and helping us fill in these dots. Looks like a lot of dots from this angle with the whole state, but as you zoom in, we do have some gaps out there. And but in other words, our mission is really, and the work we do is thinking about rare species and baseline natural communities. Where are they? How are they doing? And how can we protect them? And that is a lot of what we do. So the where are they is a lot of that super fun field work and very challenging field work we're doing in the fields, creating habitat models and going to look for species, the monitoring, and then the deliverable about how we can protect them is us delivering that data to stakeholders, the public, and decision makers, and providing expert opinion. This is an example of how our data is used. So this was on a New York Times article, I think now 3 years ago, and showing where biodiversity is most at risk in America. You could see there that Michigan isn't super red, and part of that is because we have several species that do have strongholds in Michigan, but then we have areas like Appalachia where we have the world's epicenter biodiversity for salamanders and freshwater mussels. And this is our team at MNFI. We are actually growing. So if you know of anyone looking for employment, we have several open positions for botany and zoology, both permanent and seasonal positions. And this is our team. We have ecologists, zoologists, botanists, and then a fantastic group of IT and administrative staff that help us achieve this great work. And then I just wanted to shout out a handful of our staff members who were contributors to the work I'll specifically talk about today. So for instance, Courtney Ross and Helen Anander helped me with the habitat suitability model. Eric Branch is newer to our staff, and he's been a huge help with data analyses. And Logan and Dave work with the Poweshiek skipperling, and that flies at the same time as Mitchell satyr. So we kind of divide and conquer with them focusing on satyr, and then Courtney and I focusing on Mitchell satyrs too. And I just wanted to share this photo with me and my hero from when I was, I think, maybe 20 years old. And I'll just give you a minute to read that quote. Ooh, little typo there, I see. Sorry about that. So yeah, she was an inspiration to me, and I feel so lucky that I was able to find the path in life that led me to a job where I get to work with these special animals all the time and do what I can to help protect them. Okay, so butterflies and skippers. I'd like you to all think and put in the chat. I'm going to be quiet for a second here. And what do you think I'm going to say? What are butterflies and skippers? How do you think what do you think the force of bullet points are going to say about them? I'll give you just a second to look in the chat. And I'll also take suggestions on what you think this butterfly and nectar flower are as well. And feel free to read them out as they come in too.
They all need host plants, pollinators, worth protecting.
Wow, I think honestly, in just those few, I think we hit most of them. So the first thing I put is Lepidoptera. So that is the order they're in, along with skippers and moths. So this in Greek is means scaly wings. So if you've ever touched a butterfly and gotten a little bit of dust on it, that's the scales that give them the iridescent color. Charismatic, they're part of the food web. Although with endangered species, we don't usually like to highlight the fact that they are sometimes eaten by birds and dragonflies and whatnot, but it is true, and we've seen it happen with these rare species too. They are pollinators, although it is maybe not as great of pollinators as bees and flies are, but that's okay. And I think someone mentioned this in other words, intrinsic value, and that's honestly my favorite attribute out of them, and really all of the insects and animals I work with is they're worth protecting just because they exist, and we all deserve a safe home and a place to exist too. And someone also mentioned host plants, right? So butterflies have really three primary needs: host plants, many of them nectar, but not all of them, and then that habitat. So especially I put, where are they in winter? Because unfortunately, because the monarch is so iconic and so many people study it in school, many people associate all these other butterfly species with monarchs, thinking that they just leave in the winter. And I know, speaking to this wild ones crowd, you guys are well aware that having messy gardens and yards in the fall and winter can provide habitat for them. So all the species I will talk about today are in their habitat here in Michigan 365 days a year in one form or another. And it is important to know in their life history where they are and what habit in the winter. For instance, the Karner blue pictured here is eggs over winter. And then the Mitchell satyr is in third or fourth instar larvae or caterpillars. So that's important to know as we do winter management too. So out of the approximately 150 species of butterflies in Michigan, now that's an approximate number because it does go up and down depending on, when we list things as extirpated or no longer located here. For instance, the swamp meadowlark is a really special butterfly that we think has been removed from Michigan, but we are kind of holding out hope. So out of those, like I mentioned, we have three of these federally listed species. So I'm going to start off with Mitchell satyr. And here she is. If any of you know Rhonda Spink, she helps run the Butterfly Network. This is a photo of hers. I love it. I use it all the time. Okay, Mitchell satyr habitat. So what are you seeing here? Can anyone identify any of the plants in this? We can maybe ignore that oak tree in the front because I'm standing on a bit of an esker looking at some Mitchell satyr habitat. So, and any guesses or identification of what you're seeing?
Mike
Good or
Weiss
bad?
came out with Carex stricta.
Yep,
I see other grasses and sedges, blue stem,
and these right here, anybody?
tamarack.
Exactly. Yep. So I'm going to share with you some of how we describe Mitchell satyr habitat in Michigan specifically. So we typically say that they're found in prairie fens, but the reality of it is that the natural world doesn't like to necessarily be put into these boxes that we as scientists love to assign them. So in addition to prairie fen, it's kind of a continuum of sedge meadow, shrub carr, and tamarack swamp. I often call it tamarack savanna because it's definitely not a swamp because it is groundwater fed. But so that is the important part, and also the Carex stricta, and also several other of the carices. In host plant choice studies at the Toledo Zoo done about 15 years ago, they found out that a Mitchell satyr caterpillar will eat pretty much whatever you put it on, and it will either survive or die. So they'll eat several species of thin-leaved sedges. So if anyone's aware, we have the group that has Carex stricta, Carex lasiocarpa, and then we have other carices like lake plain sedge, Carex lacustris. They only eat that first group, the thin leaf sedges. No one really knows for sure, but I have lots of theories about this, and I think that it's probably the plant fibers and the bigger sedges are a little bit too coarse for the first instars of the larvae, but who knows? And down south in Mississippi and Alabama, I think it's most likely eating Carex atlantica, and yeah. So we also have peat soil with carbonate-rich groundwater seeps. That is the fen component, right? Every Mitchell Sater site we see has seeps, and we almost always find them along streams or springs as well. And like I said, dominated by narrow leaf sedges. And then we also need a shade component. Both of the main butterflies, Karner and Mitchell's, I'm going to talk about today, both have this shade component that they need in their habitats. And it might be kind of a hedging your bet with what kind of year it's going to be. So if you put all of your eggs in just a shady area, if that ends up being a super cool, slow spring, you may be behind phenologically. Whereas if you lay all of your eggs out in the open sun, if we have an extremely dry, hot, early summer, your eggs may all become desiccated. And that again is just our supposition there. Okay, and I think this is the last of my questions for the crowd. How far do you think a single Mitchell's Sater butterfly flies on average? And while people are typing in, I'll just say this is a photo I took at Ed Lowe Foundation. Last year, or the last couple years, I've gone all around southern Michigan to good looking fens and looked for new populations of satyrs. And I think that this site, I just expected to see one right there, but they aren't there. But it's a possible reintroduction site. So, any guesses about the distance?
So there's two. One person says not far, and the other one says 1 mile.
Well, I think the not far is probably closest. So around 50 meters. And you may ask, well, did you only study a tiny little site? But no, this study was done by Szymanski et al. And they studied the largest fen habitat left for Mitchell Sater, one of the largest prairie fens in the States. And the maximum distance was 200 to 400 meters. So that's a close to half a kilometer, which is getting close to half a mile. But we do have other butterflies. For instance, if you see swallowtails or fritillaries, they will definitely fly over a mile in a day. So the one way that we actually train folks to be able to identify Mitchell satyrs, you'd think it would be looking at the eye spots on the wings to see how they're different from other satyrs or brown butterflies. But really the way we identify this is its flight pattern that's some people have called bebopping, some people have called like a drunken sailor. But it just kind of dips just below the vegetation. And if you've ever been in one of these habitats where you see that the sedges are kind of fountaining over one another, they almost have a whole other habitat below the sedges where they can be more protected from predators as well. So now we're getting into, this is an updated map. Unfortunately, we're still hoping we might find some Mitchell Saters in LaGrange County, Indiana, but it's, we'll see. So this is the Michigan map. So we have extant, meaning the Mitchell Saters are still there, and we have extirpated, meaning that they are no longer there. So I don't know if anyone's familiar with Springbrook Fen, that if you're at the Nature Center going east on D Avenue, when it changes into Sprinkle Road, we used to have a population in Kalamazoo County of Mitchell Saters right there. And that disappeared a while ago, likely because of hydrology or runoff from the road and the increase in invasive species when that road either got put in or became busier. Also, housing developments disturbing the hydrology. So this is an overview. So the work I'm talking about today is mostly in southern Michigan, some in northern Michigan. I'll get to how that works soon. And then I've also done some genetics work with some folks I'll talk about in Mississippi and Alabama. So we only have one county in Alabama where they're found in the Talladega National Forest. And then if anyone's ever ridden a bike or driven on the Natchez Trace Parkway, we have Mitchell satyrs right along that area too. So if anyone wants to go do some surveys and share data with me down there, you're welcome. Okay, so this is what I'm going to talk about a bit today. This butterfly is going extinct, and what are we doing about it? So I'm going to just touch on a couple of these. Not going to talk too much about the U.S. Fish and Wildlife Service working group that I co-lead with the service, but just know that a lot of those people in those photos and many more are all working on these issues, and we're a team. So I'm going to touch a little bit on the genetics work we're doing with the species, a little bit about habitat management, a little bit about assisted migration and the captive propagation and augmentation. That is mostly being done by our friends at the John Ball Zoo. If anyone wants to, get involved, they're a great organization and establishing new populations. So I'm going to touch on pretty much all of this, but getting into the boring stuff about how we meet all the time. And here I wanted to share with you this picture of Mitchell satyr larva. You can see here it's exactly the same color as that sedge. And if you've ever looked closely at Carex stricta or Lasiocarpa or Histocina, they kind of have like a W shape. And when the caterpillars are really small, they fit just in one of those grooves in that W. And then when they overwinter, they turn the same straw brown color as these sedges do in the winter, and they kind of go down to the base right here. This is a picture taken from the Kalamazoo Nature Center. So first off, what I lead at MNFI is annual surveys. So what I've done, because I think this will be recorded and live online, is I've removed the part where it says which site. But you can see here, this is a lot of visits to fit in. 27 visits we did in in each year, right? So 27 in that year, 37. So each year we have about 2 weeks during the flight to go do these. So this is just so you know that this is the type of data we'll collect. I will point out that if anyone is interested, we have these. In 2024, we went to this site twice. And we had 3 total observations, and the maximum count was 3. So when these two numbers are different, it means we saw butterflies in multiple visits. But this one, 2 visits, 3 and 3, that means we saw 0 in 1 visit. So for this max count to do population estimations, we are looking at that max count. How many did we see in that single day? So this line you can see is our largest population, really a stronghold for the species. And honestly, the only site in Michigan that's viable, likely to exist for the foreseeable future. But we're doing a lot of other things to help bolster some of these populations as well. And we see here that you may find this interesting, zeros here. This is a site that we are creating a new population. And then this is the type of data we can collect from them. I'm not going to spend a lot of time tonight on data, but all of that data I showed in the previous slide, we can get the lower confidence level and upper confidence level in the N, which is what we think that population was for all of the state for each year. So we can see here that 23 was a great year. And 2020, we maybe may take that with a grain of salt because of COVID, I didn't have much help in the field. I was doing some of the surveys on my own. And these data we share with landowners and all of that. So next I'm going to just touch briefly on some of the genetics work we're doing, and I put our research team up here. So Nick Haddad is a great person to work with. If you haven't read his book, The Last Butterfly, I highly recommend it. Dave runs our captive propagation facility out of John Ball Zoo. Sarah's a landscape genomics professor. They're both at Kellogg Biological Station. And Cinnamon's our postdoc that we brought on to do the lion's share of this work. And we see here we've received funding from several organizations as well. And this is the work that sent Cinnamon and I to Mississippi and Alabama a couple times to get non-lethal genetic samples from over 100 Mitchell satyrs down there so we could complete this picture. So genetic rescue to the rescue. If you haven't heard about genetic rescue, it's using genetics to help populations. The image I have on the right here is what we call the extinction vortex. So let's say one year, let's look at the top circle. One year we had a population bottleneck. That means if you think about a classic bottle with the narrow top, that only a couple species went in through the bottle to donate their genetics to the next generation. So that means that your next generation is going to have decreased variation in inbreeding. And then you have a buildup of mutation, and then you have decreased fitness, which leads to low recruitment, meaning not as many viable young. And that means a small population size. So about in 2019, we got results from work that had been done at kind of a large scale that just kind of hinted at our populations in Michigan had undergone a severe population bottleneck in recent years, and also that our Michigan individuals are closely related, or at least he thought, to those Mississippi and Alabama individuals. So we decided that we wanted to try to see if, through the genetic samples, see if those Mississippi and Alabama butterflies were indeed related closely enough to our Michigan species so that we could have them breed together to bolster the genetics. Now, when I first started saying this, I thought people were going to think I was crazy, but that research team, they were on it, and Nick said, let's bring Sarah on, and they said, let's bring a postdoc on. So then we got started. So these are, this actually, see, we still have it listed in Indiana here on this map from a couple years ago. But so we were focusing on these two populations and this population, because down here, the populations are doing pretty well. So what we did is Cinnamon created a reference genome. So she sequenced the whole genome for the species. That hasn't been done for honestly all that many animals. And then sequel whole genomes from across the range. So that means here and here and here and here. We're hoping to get Virginia samples before Cinnamon's postdoc is up. And then outcross, meaning interbreeding. I'm using my hands, you guys can't see, but interbreeding the Alabama, Mississippi, and Michigan individuals together. So right now at the zoo, we do have, with the permission of the Fish and Wildlife Service, because this is pretty aggressive conservation, this is the type of conservation, that action that you take when you've begun to run out. When I started working with the Mitchell Satyr in 2013, we had 17 populations left, and now we have between like 6 and 8. So, we've lost almost half of our populations in 10 years. So they were able to, these Michigan and Alabama and Mississippi, we were able to mate them, and they did produce viable young. However, their eye spots seem to be a little different. So we're in the middle of this work, and I think that we'll be in the next year or two sharing our results widely at as many conferences and groups as we can. So very much in progress. But the amount of work that we've gotten done since Cinnamon and my first phone conversation when we didn't know each other, 4 years ago is really astounding. So I'm really proud of this partnership, and we could give a whole hour-long talk just on this, but I'm gonna keep moving. So I have this photo. This is a Southwest Michigan Land Conservancy property, and this part is actually on private land next to a preserve. So this is a site in Cass County, and you can see here, this is the outline of where Mitchell Satyr habitat is, and this is all the dots we've seen them in the past 10 years or so. So you see that when we talk about beaver flooding being a problem for Mitchell Satyrs, or having to have these designer prescribed fire or spend all this money on shrub removal, it's, we didn't used to have these problems. We used to have wildfires or indigenous burning that could rip through miles and miles, but there would be refugia. There would be areas where the fire mellowed out. But you see here, this habitat, this butterfly doesn't have just contiguous habitat all along this area. We have a railroad track, we have roads, we have culverts, we have ag fields that have harmful pesticides and insecticides like neonicotinoids. We have beaver flooding. Beaver have made a huge rebound lately and are now flooding out some sites too. So we really have to be managing this species. It's what we call at this point in Michigan, it's a conservation reliant species. It's depending on conservationists. I'm just going to touch on a little bit. So I showed you before all those numbers, the data that we collect every year from monitoring, and I just wanted to show you another way how we use this data. So this is a private property in Cass County as well, actually. So here we see butterfly data. Each of these points is a Mitchell satyr from 2000 to 2023. So this is an current occupied habitat. And then this teal is a planned burn, a burn that they planned for 2023, right? So they are allowed to burn up to a third of occupied habitat legally when managing for a species. And that's written into a lot of the habitat conservation plan. But if we now keep an eye on this number as I open up this. So now let's just include the last 3 years before this burn. And you can see here, the butterfly has disappeared from the southern part in recent years because it has become unsuitable and too shrubbed in. So even though we consider this area occupied, we can also see that they are going above and beyond by burning this. So this is why even having, this is great data to have these point data, but even just looking at recent years, we can strategically burn this area to avoid possibly any take, which is what we call it when we harm an endangered species. So if anyone uses QR codes, I'll drop it in the chat at the end of my talk, but everything I talk about in this slide is a story map that you can find online, publicly available. It's also called Fen Resilience in Southern Michigan. So here we have a site owned by Southwest Michigan Land Conservancy, and we have 2010 to 2014. In these years, this population was booming. It was doing great. And we see here the yellow outline is the protected area that Southwest Michigan Land Conservancy owned. And we can see, wow, look at all these satyrs. They're using almost the whole habitat. And then we see what happened in 2019. We had a massive collapse of the population. We had one here. We had about, I think, 17 or 16 down here. So think back to that slide I just showed you about burning a third of occupied habitat. Imagine if they were using these points to do that burning. If they just said, okay, we know where the Mitchell satyrs are here. They're everywhere. We don't need to monitor. We don't need MNFI to come out every year. We're going to burn. And I don't know if you're seeing my cursor, but I'm circling just kind of this southern half. Let's say they said, we're going to burn a third of the habitat. And we're just going to burn all of this down here, the southern third. And they would have knocked out this, we probably would have lost this species from there. But what they did is they said, okay, well, this stinks that the butterfly is not doing well, but we thoroughly surveyed all this area with MNFI, and we're going to go aggressive and manage some habitat here. So then in 2021, they burned all of this area and all of this area. And we can see right after the spring burn. So they burned in, gosh, I can't remember, March or April. Dave Brown should be on this call to fill in. I usually rely on him. You can see the butterfly returns to these northern areas that have been burned, and we see all of these purple dots in here. So we see them responding to habitat management immediately. And again, the story map, Fen Resilience in Southern Michigan, has a time series that goes into a lot of detail if you're interested in learning more about that. So what else are we doing for the Mitchell satyr? We're looking for new populations. So de novo surveys, that's what we call a survey where we're going to a new site to check and see no known populations. So again, we see occupied, extant counties in the green and extirpated counties where we no longer find the butterfly. And so based on word of mouth from colleagues and other folks telling me where there's a good fen, or just having explored, and I made a list and we went to all these sites. Unfortunately, we didn't find any new Mitchell satyrs through all of this work. It was a lot of work that year, but we tried, and we wouldn't know if we didn't look, right? So that's one thing we've done in the past few years. Something that the Fish and Wildlife Service asked me to do was to explore assisted migration. So that is moving a species outside of its native and historic range to a new area. So this would be establishing experimental novel populations in northern Michigan. I'm just briefly going to touch on this work. So these are all the data. We tried 103 variables. The take-home message is not to read all of these, but just know we tried a lot of variables in this model to predict habitat for the butterfly in Michigan, in northern Michigan. The model we used is called Maxent, if you're interested. We also tried another model called Random Forest. The final model had just these 6 variables, and this is, you can see the percent contribution. So mean muck peat soil texture is actually our GIS geniuses invented this new variable that describes 62% of whether or not we're going to see a Mitchell satyr or have a Mitchell satyr habitat there. And so they invented this to really reflect. Remember, I said we only see them in these areas with peat soil, rich carbonate, right? So then we also had wetland, shrub wetland, and I'm not going to go into all of these, but so we made this model and this is what it looks like. So we first made the model for the current range in southern Michigan, training it with all the sites we know have Mitchell satyrs. And you can see here, not a lot of areas to choose, but we basically chose this band between Traverse City and Alpena to focus our searches. And once you zoom in, there's a lot more habitat than it looks. So in the habitat range in southern Michigan, 4% of the habitat could contain Mitchell satyr, and yeah, 1.9% suitable up here. So this is what we did is we actually went in 2 years, but in the first year, we went, checked out some sites, and then we refined the model after we realized what was wrong with it. And we focused our searches in the Huron-Manistee National Forest for the most part. Traverse City had some good habitat, but honestly, I'm worried about the pressure from development. In the sites that looked like they had good fen, there was nearby developments and golf courses that I think would disrupt the hydrology in the long term. So these are all the sites that we thought were good enough to potentially be Mitchell satyr reintroduction sites, or introduction sites, I should say. And you don't have to read this whole thing, but we have a report I'm happy to share with everyone. We don't have it publicly available yet because I need to redact it and find the time to do that. But this is one of our top scoring sites. So here's the description we wrote about that. And again, drop me a note if you are really interested in learning more about this. So moving on now to the Karner blue. There she is, beautiful, 2 mating Karner blues. All right, now I'm going to exit out of this. Oops. End presentation, how do I do that? Oops. There we go. I'm going to play a short video for you all that I made with the Michigan DNR this year to celebrate the Michigan, actually, I'll let the video speak for itself and be quiet for a minute.
Welcome to Allegan State Game Area, where a tiny butterfly unites partners for conservation. It's here, along the winding Kalamazoo River, that we find widespread grasslands and towering oak trees that comprise some of our state's best savanna ecosystems. Many species call Allegan State Game Area home, including the Karner blue butterfly. These butterflies are charming and beautiful, and despite their small wingspan only being about the size of a postage stamp, they have had a big impact on conservation in Michigan and beyond.
Allegan State Game Area is a stronghold for Karner blue butterfly populations. The Karner blue serves as a crucial flagship species here. By managing oak savanna communities at Allegan to support Karner blue, we observe benefits to many other wildlife and plant species that share this space.
The Karner blue butterfly has one host plant called wild lupin or sundial lupin. The butterfly is inextricably linked to this plant, as the caterpillars must eat it to grow into an adult butterfly. This lovely perennial is native to Michigan and thrives on sandy soil, which is abundant here in Allegan. With shared knowledge, resources, and dedication, we can make significant strides in habitat restoration and increase public awareness of the incredible biodiversity around us. To ensure that Karner blue butterflies continue to thrive in the Allegan State Game Area, we must maintain an open savanna forest as well as the sundial lupin growing in the understory. To achieve this, we use two management tools that are key to maintaining and restoring these habitats: prescribed burning and forest mowing. Fire stimulates lupin growth, while mowing reduces woody encroachment from crowding out the lupin.
Partnership is the cornerstone of success in this endeavor. Joint efforts between DNR Wildlife Division and the Michigan Natural Features Inventory are underway to monitor and research populations and maintain suitable habitats critical to the Karner blue butterfly's survival.
This year marks the 50th anniversary of Michigan's Endangered Species Law, a milestone highlighting decades of dedicated efforts to protect and preserve the state's most vulnerable wildlife.
One emerging threat to Karner blues is a species called bigleaf lupin, which is native to the western United States. Bigleaf lupin is used as an ornamental plant in landscaping because of its showy blooms. The Karner blue can mistake this for its host plant, but the caterpillars are unable to eat it when the eggs hatch. For residents of the upper Midwest, it is imperative to plant native sundial lupin, as invasive bigleaf lupin can escape gardens into Karner blue habitat.
By incorporating native plants into your landscaping, purchasing a wildlife habitat license plate, or a hunting license, you can play a critical role in conserving Michigan's natural heritage.
We will continue to work.
Okay, we don't need to see more of me talking right now. So I wanted to share that video because that's honestly a lot of what I would have shared to describe the habitat of the Karner blue butterfly. And I'm proud of the drone footage I shot for that video and wish they would have left my face out, but okay. So we touched on this a little bit. From our website, if you go to MNFI's website, we have all species accounts of all of these rare animals, including county level maps on where they occur. And if you hover on each of these, it'll tell you what's the last year we saw each other. So like we mentioned, the species is monophagous. It only eats one species, sundial lupin. And that shade sun dynamic, similar to what I talked about with Mitchell's satyr, but the Karner blue sun and shaded lupin is much more well studied. And a lot of those studies took place in northern Indiana, from where the species is now extirpated, and Michigan. So for this species, since it lives in these dry, sandy barrens habitat, the sun and shade lupin is really important because this butterfly, unlike the Mitchell's satyr that has one generation per year, the Karner blue has two. So when that second generation is flying in later summer, kind of mostly through July, the lupin that was lush and green for that first generation in full sun, that lupin is toast. And the shade lupin then is what they use to eat more so. They need nectar sources, plus ants, and I'm going to touch on that in a second too. So this is an image I took from some folks that did this work out of Indiana Dunes. Like, unfortunately, a very hot year a few years ago led to the local extirpation of that population. So here we're seeing midsummer, some of that full shade lupin, right? And this is our current climate scenario. We have two of these flights, and we have lupins starting to go brown right here. With a +2 or +4 degrees Celsius climate warming scenario, the butterfly enters a third flight where the lupin is done for the year. They likely go extinct. With a +6 degree climate scenario, this is Celsius again, we may have even a fourth flight and local extinction. So we're also in the very, very early stages with Fish and Wildlife Service about talking about the same sort of assisted migration to northern Michigan. One thing we don't have in northern Michigan is lupin. So we are, along with Lars Bredvig of MSU and Tyler Bassett, we just put together a proposal to look at planting lupin in northern Michigan in preparation for this and under kind of experimental conditions. So Yeah, I was going to ask another question, but I think I'll just keep moving in the sense of time. So, Karner blue and ants. So, they have a facultative mutualism, which means that they benefit from each other, but they don't need each other. When you read about kind of the yucca moth and its host plant, they have an obligate mutualism where they need each other to survive. So, what happens is these ants feed on this honeydew-like secretion glands that the caterpillar produces. And in return for that favor, the ants will protect the caterpillars. It's sometimes even taking them down into their ant mounds. And oh gosh, you guys, I'm sorry, my dog just farted. Get out of here. Whew. Sorry, they will take them and protect them in the ant mounds. So we often find I took this photo at Allegan State Game Area. We often found Karner blues. This is what you need to manage for Karner blues: lupin in sun, lupin in shade, ant mounds, and nectar plants. I'm going to touch on a little bit nectar sources here. So this is what our protocol looks like. I'm going to go through this a little bit quicker because I got into storytelling mode. So we use a survey 123 for data collection. And last year we always mark what they're doing, but this year I added a conditional form that if we selected nectaring, we would then type in what they were nectaring on, right? So yeah, I'm just gonna play. Everybody know these two species? On the left, well, we had, spotted bee balm, right? Or horse mint, some people call it. And then this is world milkweed. And I just want to say that this is one of the coolest nectar sources. It is like the butterflies love it. So we studied all these nectaring, and I just wanted to shout out some other species that also nectar. So here is the relative abundance. So the most common species we found that were nectar sources: St. John's wort, spotted knapweed. Invasive, right? They were the most abundant. So you'd think, isn't a butterfly going to use the most abundant species? But if we look here, we see that even though St. John's wort was the most abundant species we found, that we only found 8 nectar observations for a total of 9 individuals. That means one of those observations had 2 butterflies at once. Spotted knapweed was used a decent amount, but if we look here to our species down here, we see that even though world milkweed was the 12th most abundant, not very abundant, host plant, we had 23 nectaring observations with 73 butterflies. That means that each time we found world milkweed, we found an average of 3 butterflies using it. That means that that's a limiting nectar source. So that is us providing data to those land managers saying this is a host plant that they are overutilizing based on its abundance. And the same story here with butterfly weed. And also everybody heard the news in December about the monarch listing. So this is a great way that we can benefit multiple species by putting the right species of milkweed in the right habitats. So that's an example of how MNFI, we changed our data collection to address the needs and the data that land managers. We work really closely. We're not over there in our research facility just giving land managers whatever we think is important. We're listening to them and we're changing what we do and how we do it. And this is
Actually,
just
we
showing
had
that data.
a question about the Karner blues, which I'm curious about too.
Yeah.
Do they also only travel short distances?
Absolutely. We do find with Karners and many other butterflies, the males are the dispersers. So males tend to kind of go off into unsuitable habitat looking for new patches. So yeah, and I could talk for hours about this species, but they have they live in early successional habitats, meaning that the habitats will naturally close in and go to forest, these openings where they live. So they're constantly moving and looking for new habitats, but typically only a couple 100 meters in their life. And if they have excellent habitats, especially the females with the ant mounds, the shaded lupin, the sun lupin, and the good nectar sources, those females aren't likely to leave, right? If you were ready to have a baby, would you want to go and just search out, out in the wilderness and hope to find a home by the time you're ready to lay your eggs, right? So those males tend to kind of wander. So yeah, good question. And I just put in here, it's an umbrella species. That's an ecological term that when we protect for one species, we're creating this umbrella of protection for all of these other species too. So we have a lot of plans left for this species, like I mentioned, assisted migration. And I'm just really quickly going to touch on the Poweshiek and then I'll be done. So historically, this is where I'm from in Iowa. Unfortunately, the county that it's named after, Poweshiek County, was one of the first it became extirpated to the same time that industrial agriculture as we know it today began. And now we've lost it from pretty much all but 5 locations. So I think we went from like 300 or so known locations to about 5 here, including 2 in southern Michigan. And this is just another example of how fire and butterflies interact. So here we see some reared butterflies from the zoo released here, the black ones. And then we see them entering right after they did that spring burn, going to use that habitat. And you see here, again, that use of data to know where to strategically burn. They burned right up until where the wild butterflies were. So finally, I know I'm talking to wild ones folks, and the number one way we can help all of these butterflies I'm talking about, plus all of the common and important species, is planting native. And every little bit of curb lawn or garden helps getting your seeds from a reliable source. And I showed that video, so I mentioned this. On our left, we have our native sundial lupin. On our right, we have the Lupinus polyphyllus or big leaf lupin. It is a native, all species are native somewhere, but this one is not a host plant for the Karner blue. The chemoreceptors on the female's feet, if you ever see a butterfly land and it's kind of lifting its legs up and forth like this, it detects that it's lupin because it's chemically similar enough. But then the caterpillars hatch from their eggs and they begin eating and they can't complete their life cycle. They will die on that. And I find this up in northern Michigan more than around here. But really, if you want to help spread the word, you can show that DNR video that I showed, or just let folks know that this is honestly a textbook reason on why, example of why to plant native. And, this is a great lupin species. Out west, they have tons of lupin species, right? We only have the one that our Karner blue co-evolved with. And this is, again, if you can take a quick shot of that QR code, that will take you to share all about how you can submit data to MNFI to help us do our work. And we will give you some sort of award if you help us find a new population of Karners or Mitchell satyrs or Poweshiek. I would love to have that happen in my career. So. And we also have a local artist, Quercus Creative, out of Grand Rapids. He recently made these stickers for me, and they're only up for sale for another couple days. So if you go to Quercus, he's doing a pre-order, and you could get some of these butterfly stickers. He also has a prairie fen line that he did for us as a fundraiser a couple years ago during COVID when we couldn't all be together. And with that, I will end. Oops.
Fantastic, Ashley. That was fascinating. We do have 8 more minutes left until 7:30. So if there's anyone else who has any questions, you can put them in the chat and I certainly can ask Ashley. We'll give them another second. What's your naughty puppy's name?
My puppy is Honey. This is her.
Honey?
She's a Pomeranian Chihuahua rescue.
Very cute.
Yeah, she sits on my lap while I'm giving talks and then she farts.
Support animal. Let's see here. Mike Weiss asks, drop seed is a host for the Poweshiek, so does it help to plant it in our gardens or does it need large colonies?
Oh gosh. Well, unless you're in Oakland County, the Poweshiek isn't going to find those, but prairie dropseed is a great plant. And yeah, that is something to keep in mind when you do plant these host plants for butterflies, especially a larger butterfly like a swallowtail that's going to eat dill or something like that. You need to keep in mind that a single caterpillar can eat a ton. So dropseed and mat muley are the two plants that we think Poweshieks use. And we honestly, we find them, we think they use mat muley, but we're not sure. So Mike, plant the drop seed, but maybe we'll find some Poweshieks to populate Southwest Michigan again. I'm always the bearer of bad news. I'm sorry. Do we collect data from iNaturalist? We do use that occasionally, especially when we have a species that's just added to the endangered species list, so we don't have years of data, and then we can QC that. But for especially the species I spoke about today, we aren't really finding any, of the really rare species, but we do use that when we have limited data. Do we have an idea when the populations of these butterflies really started dropping off? Does it coincide with other known species losing populations as well? So yeah, the grass skippers like the Poweshiek, the rise in neonicotinoid, Roundup Ready, corn and soybeans coincided with the, and also farm bill changes coincided with the drop of those skippers. And there's a thought that too, the Poweshiek nectars a lot. The Mitchell satyr doesn't nectar so much, and that these plants, the chemicals become concentrated in the nectar. So that's why we've seen, for instance, Poweshiek disappear way before a Mitchell satyr in a site where they were together. Yeah.
Looks like that's the last of the questions. So thank you, Ashley, very much for joining us on our first program of 2025. Obviously, it's winter, and so we don't have a ton of Wild Ones activities going on, but for the month of February, our Pints and Plants at Brewery Outre, downtown Kalamazoo, is on Thursday, February 20th. And we'll be down there starting at 7 p.m. to hang out and talk plants, maybe butterflies this next month too. Our February program is on Wednesday, February 26th, starting at 6:30. It also will be on Zoom. We are having William Kirst of Adapt Community Supported Ecology. He is going to be talking about garden design from the ground up, using your native soil to inform native plant choices. So we have, in addition to that, a lot of very exciting speakers lined up for 2025. So we hope you'll join us again. Really quick, Ashley. Oh, I see you're typing. Would you also add your email in there so people can reach out to you?
Okay.
If you're on Facebook and you liked the event, I will add that information on the Facebook page, and we can ask our web person to add it as well, and that MNFI QR code.
Thank you, guys.
So Cole-Wick at msu.edu. Thanks so much for joining us tonight. Have a great evening.